Hello everyone. In this video we will cover eukaryotic or human cell. We call it eukaryotic because it has nucleus. Some cells don't have nucleus, then we call them prokaryotic. So let's look at this model and identify major organelles of a human cell. So we're going to start with the cell membrane. So cell membrane is the outer covering of the cell. So that this part is cell membrane. Now cell membrane or plasma membrane separate external environment from cytoplasm of a cell. The biggest organelle inside is nucleus. Now all the area between cell membrane and nucleus is called cytoplasm. Cytoplasm includes fluid portion. This is fluid portion over here, or aqueous portion, that is called cytosol, and organelles. So all these organelles over here. So cytosol and organelles make cytoplasm. Cytoplasm is everything between cell membrane and nucleus. Right now, the biggest organelle is nucleus. Nucleus has several parts. This white part is nuclear envelope that separates fluid and chromosomes that are inside the nucleus from the rest of the cell, from cytoplasm. So nuclear envelope has pores. You can see those little holes. Nuclear pores allow molecules and compounds and ions move in and out of the nucleus. Now this fluid part of the nucleus is called nucleoplasm, and this yellow part is nucleolus. Now if you can see these blue structures right there, over here, those are chromatin. Right, so function of nucleus is to house DNA. Chromatin is a DNA. It's made of DNA and proteins called histones and a little bit of RNA. Now nucleolus is the place where ribosomes are assembled and ribosomal RNA is synthesized. Now, what are ribosomes? Ribosomes shown here with these white dots. So here, so you can see dots right here. So those are spherical, pretty small organelles, and function of ribosomes is protein synthesis. You can see ribosomes are found in the cytoplasm, but to make ribosomal RNA, because ribosomes are made of ribosomal RNA and small proteins, to make ribosomal RNA and assemble ribosomes, we need nucleolus. Right, so let's review what we covered so far. Cell membrane, cytoplasm, cytosol, fluid part of the cytoplasm, organelles, nucleus, nuclear envelope, nuclear pores, nucleoplasm, nucleolus, and chromatin that is made of your DNA, and ribosomes, those little, I cannot see it here, little white dots on this model. Now this cell has only one nucleus. So this is uninucleated cell. Most of cells of human body are uninucleated with one nucleus. Some cells have more than one. For example, your skeletal muscle cells, and we call them multinucleated. Some cells, like red blood cells, they don't have nucleus, and we call them anucleated. but most cells in a typical human cell would have one nucleus. Now, this model has only one nucleolus, but sometimes we have several nucleoli inside the nucleus as well, right? Okay, let's look at another organelle. So we already mentioned ribosomes, those dots here and here. And you can see ribosomes are attached to another organelle. This is RAF-ER. So RAF-ER is a membranous organelle shown here in kind of light green color. Right, so here's the ribosomes attached. And this blue part is fluid inside the lumen of RAF-ER. So RAF endoplasmic reticulum. The job of RAF endoplasmic reticulum of function is to help in protein synthesis. It's a side of protein synthesis. Also, when proteins are synthesized by ribosomes, they deposit it inside the lumen, and within the lumen we have folding and modification of newly synthesized proteins. Right. Another structure shown right here, you can see it's similar to RAF-ER. The difference is no ribosomes attached. Or this part over here, if we don't have ribosomes attached, we call it smooth ER. Because we don't have ribosomes, we cannot synthesize proteins. So function of smooth ER would be lipid synthesis and lipid metabolism. So for example, you would synthesize cholesterol or phospholipids inside the lumen of smooth ER. Also, some ribosomes are free ribosomes. They're not attached to RAF-ER. They're not shown on this model, but keep in mind you might have ribosomes that just inside cytosol. Next organelle we want to cover is over here. This is Golgi apparatus. So when ribosomes make proteins or lipids are synthesized inside smooth ER, they are packaged inside vesicles, and those vesicles move to the Golgi. This is receiving side or cis, and this is trans side of the Golgi. Now Golgi will continue modification of proteins and lipids. And when it's done, proteins and lipids will be packaged in these Golgi vesicles right here, or secretory vesicles, and transported to their final destination. So Golgi will receive, sort, and package, modify and package proteins and lipids, receive them from RAF and smooth ER, and package inside secretory or Golgi vesicles and send them to final destination. They can be secreted out of cell, or they can be incorporated in a cell membrane, or even incorporated into lysosomes, for example. Right? So by the way, let's cover next one, that is lysosome. So here's a lysosome over here. Okay, let me move so I have light again. There we go. So lysosome shown here in yellow color. So for example, this one right there, or over here, right there. Lysosomes, they have digestive enzymes inside them. So it's a spherical organelles that have digestive enzymes, and what lysosomes do, they can fuse with another vesicles and digest different maybe bacteria that enter the cell, or organelles that are not function anymore. For example, if you have one organelle and it's not functioning, then lysosome will fuse with the organelle, release digestive enzymes, and break down those non-functioning organelles called autophagy. Or if it's a white blood cells, it might engulf the bacterium and lies inside the vesicle, and lysosome will fuse with the vesicle and digest the bacteria, right? Anyway, lysosomes have digestive enzymes. We call them hydrolytic enzymes that break down proteins, glucose, nucleic acids, lipids, so on, right? So when you need to get rid of something inside the cell, you will digest it using lysosomes. Now another vesicles are dark one, like this one, or shown here. This is peroxisome. They have enzymes too, oxidases and catalases. Lysosome important in fatty acid metabolism, and also they help you to get rid of free radicals. Free radicals are formed when peroxisome break down long chain fatty acid, brain chain fatty acid, so fatty acid, right? So they break down those fatty acids. They use catalase to do it, but then you create lots of free radicals that are dangerous to your cells. They can damage your DNA, can damage organelles. So peroxisomes will convert free radicals into water and oxygen. That is not toxic to your cell. Right now, this organelle over here is a centrosome, and inside we have centrioles. Now these centrioles will replicate, and when cell is ready to divide, centrioles will move towards the opposite poles of the cell, and they will form the spindle that we call mitotic spindle. So they are important in the cell division, in a division of a cell. And now the last but not the least organelle shown here, because if you need to synthesize proteins using ribosomes, you need to make lipids using your smooth-CR, you need to package proteins using rough-ER, you need to process, receive, package newly synthesized proteins and lipids using Golgi. Then you need energy to do all these processes. And here mitochondria are organelles that gonna provide energy for all those cellular processes. Mitochondria is plural. Mitochondrion is singular. So here you have several mitochondria. They will synthesize ATP. Now some cells have lots of mitochondria if they require a lot of energy. For example, your skeletal muscle cells, your heart muscle cells. Some cells might have fewer mitochondria, like your skin cells, but all cells except of red blood cell have mitochondria, so they can make ATP and carry on their cellular functions. Okay, so very quick review. Cell membrane or plasma membrane, nucleus, between nucleus and cell membrane, cytoplasm, cytosol and organelles, nuclear envelope, nuclear pores, nucleoplasm, nucleolus, chromatin, ribosomes, those dots here. I don't know if you can see them or not. I'm not very professional camera lady, so that's the ribosomes. Organelle that ribosomes are attached to is rough-ER. Similar structure but without ribosomes. Smooth ER, lysosomes, peroxisomes. So here's the lysosome, peroxisome, centrosome with centrioles, and Golgi apparatus, secretory or Golgi vesicles, mitochondria, and blue part, just because it's so pretty, nice, and bright blue, I think I should mention it. This is a lumen of either rough or smooth ER. This is look like smooth ER because I don't really see ribosomes around it. Yeah, so if you don't see ribosomes, oh, I forgot, this might be some vesicle or vacuole. I think this is a, you would say, some vesicles. So this cell is taking something in, maybe pinocytosis, maybe some fluid, maybe right, something else. But here is a part of rough ER. I wanted to show you a vacuole, but I don't see it on this model. So yeah, I don't think we have vacuole. Anyway, that's it for this video. Thank you for watching, and I hope it was helpful.